In my lab, we focus on somatic stem cells, which are a small population of cells that in fact is found in many different tissues of the organism, and they are very central to maintain the homeostasis of the tissue they reside throughout the lifespan. Because when the tissue is under stress or is damaged or require some particular new regeneration, stem cells have the ability to provide this. They are engaged and they can self-renew and differentiate to maintain the tissue, to repair it, to regenerate it. And they are very critical also during aging, because like other cells, also stem cells do age and lose their capacity to regenerate the tissue. I investigate the biology of these, how stem cells regenerate and lose this capacity over time, with the idea that this is something that can be understood first and improved later, so that the tissue can be able to regenerate also at older age, and in this way contribute to extending the health span and the lifespan of the organism. Somatic stem cells function declines with aging, and this might be happening in very different ways depending on the tissue we are talking. For example, in the skeletal muscle, the number of stem cells decreases and also their activity is less regenerative, and this altogether contributes to a loss of regeneration of the skeletal muscle, and for example to sarcopenia in the elderly, which is a very important condition that impairs the quality of life and increases the frailty of elderly people. In another tissue like the hematopoietic system, hematopoietic stem cells increase in number with aging, but again they regenerate the tissue less efficiently. For example, they tend to do more myeloid cells and less lymphoid cells like B and T cells, and this is very well established to contribute to a phenomenon that is called immune senescence, that for example was very important during the COVID pandemic, because most of the elderly people have really difficulties in cope with viral infection, and this is because their T cells and B cells are less able to respond to this kind of stress. And this all depends on hematopoietic stem cells that constantly generate this kind of cell within the tissue. Stem cells in general hold great promise for regenerative medicine, and there are many applications that are currently under investigation. Only a few, unfortunately, reach clinic, but there is very intense research nowadays here in Bellvitge, but in also many other places worldwide, that will for sure end in an increase of regenerative medicine strategy for the clinic. In my case, we are hoping to push forward the possibility of increasing the regeneration of tissue like the hematopoietic system or the skeletal muscle for elderly people, with the idea of being able to increase their health span, so the period of life in which they live in a healthy status, and also overall the lifespan of the patient. Normally in the lab we work a lot with mouse model. We isolate, for example, hematopoietic stem cells from the bone marrow of the mice, and we isolate this small population of cells. We look at them in vitro, we profile them by single cell sequencing or by microscopy. We analyze a specific marker of expression, and we screen drug treatment to improve their capacity of dividing and regenerating. to address whether these treatments are really working also in vivo, we are able to re-inject the cells into a new mouse and measure how do they perform, if they improve their reconstitution and regenerative capacity, their lymphopoiesis after the treatment or not. And in this way, we have been able to conduct several studies on lifespan and health span on the mice. And what we hope to continue and push forward now is something similar with human and patient sample. So we have some open ethical permission to analyze human bone marrow samples and perform treatment on human cells in vitro and analyze how do they respond at the treatment, if they improve their regenerative capacity. And right now, we measure, in the case of human cells, their activity by performing xenotransplantation. So we re-inject human cell into mice that serve as carrier of the cell and allow us to measure the activity of the human cell. And these are studies that are done in a kind of preclinical setting, and if successful, we hope in the next future to move it forward to more clinical studies. I did choose to come here because of this newly established program on regenerative medicine. I saw here a possibility for some mainly basic research to have the possibility to meet more translational research, and this is one of the strongest pillar of this new program, to bring together basic research on stem cells, that is my field, close to translational and clinical need. And the idea of the program and the way it was pursued was for me a great possibility to move forward also for me personally in my career and in my scientific development. So yes, it was a choice, and I am very happy with it. I think it's meeting my expectation. I feel like here I have many connections, especially with clinicians, and the possibility to really develop my science more in this direction. And I'm really happy about the scientific community, and again the possibility of really exchange idea and science with different personalities and also clinicians. One of the advantages of this kind of work is that you can be really flexible. Most of the time is about following an idea, performing experiment, and it can be time consuming or requiring some dedication, but it can be organized in a flexible way. And this allows me in many situations to couple my work and some intense days of working with some more easygoing and dedicate myself to the family or to activities in the free time. And this is definitely an advantage of this work. I definitely would like to keep doing great research and to increase further my visibility in Spain, in Europe, and possibly worldwide, and to keep working with fantastic scientists. One of the privileges of our work is that you can build connection and really teamwork with great scientists, and I hope that I will be growing much in this direction.